Is the screen visible to all of you?
Yes, sir.
Okay, very good. Okay, so in the last class, we had done some molecular experiments, the mixture transforming principle, and also Martha and Chase experiment that tells us that DNA is the genetic material, right? So who was present in the last class? I think Fatima was there, right? Fatima, you were there in the last class on Monday? Yes. Fatima, would you like to give me a brief recap? Am I audible, Fatima? From here. Fatima, am I audible to you? Yes or no? Some answer will help. Atif, am I audible to you? Yes, no. Am I audible, people? Yes. Okay, Rifat is saying yes, I'm audible. Can anyone speak? Rifat, Fatima, Atif, for a recap of the last class?
Sir, can you hear me?
Yes, Atif, I can hear you now. Okay, very good. Would you like to start with the recap, Atif? What we did in the last class?
1 minute.
Yeah.
Okay, sir. So we learned about the experiments to find genetic material. So first we learned that Meister discovered nuclein, that is the substances inside a nucleus. Then we learned about the transforming principle, which this was found through a series of experiments which were done by Frederick Griffith in 1908. He used the Streptococcus pneumonia bacteria, and then he took two strains, that is a smooth and shiny S strain and a rough R strain. So he injected both these strains in mice, and he found out that the mice died in the S strain and they lived in the R strain. Then what he did is he heated the bacterial strains, and then when he heated the S strain and injected them in mice, the mice lived. The mice lived because the heat killed the S strain. But he found out that when he combined the live R strain and the heat killed S strain and then injected in mice, he found out that the mice died. So through this, he concluded that there must have been a transfer of genetic material and hence a transformation from the a transformation by the R strain to the S strain, because he found a recovered S strain in the dead mice.
Yes, right. Beautiful.
Then we learned about the purification of biochemicals. So scientists
but
purified
this
everything.
this is in continuation. So he figured out that something is being transferred, something which is the genetic material carrying these genes of virulence or lethality are being transferred. That's there. But this experiment did not tell us what is that thing, what is the chemical nature of that thing. So the next step was obviously I ask you, how will you figure out which component of biryani you are allergic to? Remember, Atif? And it was your
Yes, sir.
answer only that we can check one by one. Yeah. So this is what is the second experiment. Yes, continue.
Yes, sir. So the continuation of the Frederick Griffith experiments was that scientists purified all the biochemicals from the heat-killed S strain to find out which one was the one that was being transferred to the R strain. So yeah, they purified all biochemicals and each component they transferred to the R strain, and then they found out that DNA
One
was
by
the one
one.
which was responsible. Yeah, they transferred one by one, and they found out that DNA alone was responsible for the transformation of R strain into S strain. But
Yes.
still, some biologists were not convinced by this experiment. So next, Alfred, Hershey, and Martha Chase performed the experiments, which gave the unequivocal proof that DNA is genetic material.
What was that experiment?
So yes, so Hershey and Chase took some bacteriophages, which are viruses that infect and kill bacteria, and then they performed radioactive labeling by culturing these bacteriophages in. They culture one batch in radioactive sulfur and another batch in radioactive phosphorus.
Yeah.
So when one batch was cultured in radioactive sulfur, it was found out that the protein code was labeled due to the disulfide bond in it. And in the other batch, the DNA is labeled due to it having the phosphorus. So
Yes.
both these
And both
bacteriophages were
these
then.
components are exclusive. Sulfur is exclusively present in protein, and phosphorus is exclusively present in DNA. Okay. Yeah.
Yeah, okay. So yeah, both these batches were made to infect on E. coli bacteria, and then so yeah, both of them injected their DNA to the both of them transfer their DNA to the bacteria, and then the the protein coat was agitated through a process known as blending.
See,
Which,
at this point of time, we do not know did they transferred DNA. So you are making a statement because we know it's the DNA. But remember, this experiment is being done to prove that it is DNA. So you should write, or you should say that when we allow the bacteriophages to infect E. coli, then it transfer its genetic material to the E. coli in order to infect it, right?
yes,
If you
sir.
already say that they transfer DNA to the E. coli, then the whole point of this experiment is no longer valid, right? It's not required because you know that it is DNA. How will you figure
Yes,
out through
sir.
this experiment that it is DNA? This is what you have to explain. Yes, very good. Come on.
Okay, sir. So after the genetic material was transferred, then
Mhm.
through a process known as blending, the phages were agitated, and then this caused them to release from the bacteria. Then the then the bacteria and the phages were uh, spun at high speed, which
Mhm.
is the process known as centrifugation. And then in the first case, um,
Where the where they
yeah,
were
in the
grown
first,
in the first case where
uh,
the protein was labeled.
yes, in the first case where they're grown in the radioactive sulfur and the protein coat was labeled, when they're centrifuged, the supernatant,
Again,
there is radioactivity
yeah,
in the supernatant, and in the bacterial cells, no radioactivity was detected.
yes,
And then in the second batch, which was grown in radioactive phosphorus, radioactivity was detected in the bacterial cells, and the same was not detected in the supernatant, which is the phage coating. So through this, we can conclude that DNA was the material that was transferred from the uh, phages to the bacteria, and this gives us an unequivocal proof that DNA is the genetic material.
yeah.
Yes, sir.
Then the last thing we did
Then
was this question.
then we describe the properties of genetic material. So we learned that genetic material must be able to self-replicate. It must be it must be chemically and structurally stable so that it will not affect other generations. It must allow slow changes that drives evolution and variation, and then it must be able to express itself in the form of Mendelian characters.
Yes, so this point that they should be chemically and structurally stable, this is actually proved by the first experiment done by Griffith. So if I ask you, when Griffith heat killed the S strain, which means heat killed the bacteria, right? But heat did not destroy the genetic material, because even after heat killing the bacteria, something could pass on to the R strain and can still work. Yes or no?
Yes, sir.
Yes. This proves that even after killing a bacteria through heat, DNA has the genetic material, which we now know after the Martha Chase experiment that it is DNA. It has the ability to stay stable and remain intact and functional even after applying heat. Yes?
Yes, sir.
Yes, very good. So what are these four properties? Able to self-replicate, okay, chemically and structurally stable, it should allow slow changes and express itself in the form of Mendelian characters. Very well. Now, when we come down to these four characters, and when Martha and Chase proved that it is the DNA, if I ask you that can RNA be the genetic material as well? Can RNA, because RNA also has the same properties as of DNA. It has phosphorus. So if you would have labeled a phosphorus in an RNA, there are many viruses that have RNA as their genetic material. So yes, RNA can also be a genetic material. Is it clear? And this is where I ask you to go back and read these stuffs. Did you go back and read this from NCERT? Did you do the homework, everyone?
No, sir, I was not able to. I was traveling yesterday.
Okay, okay. So then you have to discuss with me. Others, please pay attention and let's discuss this thing because it's important. So we now know that DNA is the genetic material. But before that, let's understand some more points. Now, having said that these are the four characteristics, there are three potential candidates: protein, RNA, and DNA. Protein fails at the first stage only. Protein is unable to self-replicate. Protein relies on RNA to replicate, yes, or to make its copies, like not basically replicate, but new proteins can only be formed when the RNA is there and it is read by the ribosomes, and then protein is formed in the cell. Correct? Yes or no?
Yes, sir.
Now we are left with DNA and RNA. Now the question is, which is a better genetic material? Write down. Which is a better genetic material? DNA or RNA? What do you think? Which is better? DNA is better or RNA is better? Let's go one by one. So about the point of replicating, both are able to self-replicate. RNA can also be formed, DNA can also be formed. So, but when it comes to chemical and structural stability, DNA is more stable than RNA, right? Now it has
Yes,
two
sir.
reasons. Now write down first thing. DNA is more stable than RNA. More stable than RNA, as it is double stranded. As it is double stranded, and the two strands are complementary. They are bound together. So this binding of the two strands together protects the DNA from external, damage to a great extent. So a lot of chemical reactions DNA cannot participate in because it is well protected, covered, and double stranded. But RNA is more reactive because if you imagine two strands, so if there are two strands, let's see, these are the two strands. Okay, imagine in between there are bases. Okay, there are, at every step there is a base which you cannot see. So this double stranded structure is protecting it from attack of the chemical moieties from both the sides, right? Because everything is bind to something. You understand? It's a complete. If I remove one strand, now there is only one strand. So whatever is protruding out of that strand, it's open, right? It is exposed to the environment, correct?
Yes.
It is not covered by something. So RNA has a hydroxyl group at the second carbon. Okay, so DNA is more stable than RNA. RNA, write down, RNA has, because it's single stranded, 2' hydroxyl group, 2' OH. So at the second carbon there is a OH group present in every nucleotide. This group present in every nucleotide. Which makes it more reactive. And easily degradable. Okay, so because of this group, mRNA or RNA is more reactive, which means less stable. And something which is less stable is easily degradable. You understand, everyone? Is it clear? Now write down. But this property of being more reactive also give an advantage to mRNA. What is the advantage that RNA has and DNA does not have? So DNA is more stable, less reactive. But if something is more reactive, is there any advantage there also? RNA can catalyze reactions. It is catalytic. Do you understand?
Yes.
RNA is catalytic. Okay. So it is chemically more reactive, structurally less stable. So write down. RNA is catalytic, chemically more reactive, structurally less stable, and hence has the potential to act like an enzyme. What does enzyme do? Or what do catalysts do? Yes, and what is a what are catalysts? What they do basically?
They increase the rate of reaction.
Exactly. They make certain reactions happen which was not possible or feasible at that particular temperature, right? For example, for the breakdown of glucose in our body at normal temperature, we are doing oxidation. For oxidation, we need, so oxidation is like combustion reaction, right? Where we are burning something or breaking down something in the presence of oxygen. Now that happens at a very high temperature, burning, right? But when we are doing the same kind of reaction in our system, in the cells, we are doing it at 37 degrees Celsius. We need certain enzymes for glycolysis to happen. Now, if RNA has this ability to act as an enzyme, then these RNAs that act as enzymes are called ribozymes. What are they called? Ribozymes. So this is the benefit that RNA has over DNA. RNA can act as an enzyme, but DNA is very, very stable, so it stays as it is. It does not participate in a reaction. It does not make any reaction happen. So if I have to summarize in one line, RNA is chemically reactive, structurally unstable. But DNA is chemically less reactive, but structurally more stable. Correct?
Yes, sir.
Yeah. So about DNA, write down. DNA is chemically less reactive and structurally more stable. So if you have to store information, for example, anything that we have to store for throughout our life. So for storage of the information purposes, which is better, DNA or RNA?
DNA.
Yes. So write down. Therefore, DNA is better for storage of genetic information. Okay. DNA is more preferred for storage of genetic information, and hence is a better genetic material. Okay. Now there is one more catch here. RNA is unstable, so it will also mutate faster, right? Because mutations
Yes.
are something that are sudden alterations in the sequence. If something is more stable, then the alterations are difficult, or it will not happen very frequently. But if something is less stable, these alterations can happen more frequently. So RNA is less stable, so it mutates at a faster rate. Keep writing. RNA mutates at faster rates. Therefore, viruses which have RNA as the genetic material, viruses which have RNA as their genetic material will mutate and evolve faster, will change and evolve faster. Is it clear? Is it clear, everyone? So that's why
Yes.
certain viruses are not easy to contain if they are RNA viruses. Okay. Now talking about translation part, you know that RNA, if any organism's genetic material is RNA, there is another advantage to it. The disadvantage is that it is unstable. Okay, that's there. But if you somehow are lucky to go through it and stay intact and evolve faster, then you do not have to depend on the process of transcription, because RNA can directly get translated in a protein. Yes or no? Whenever you need a protein, you can directly access the RNA and make proteins. You do not have to make from DNA a copy of RNA and then from RNA a protein. But DNA depends on RNA for protein synthesis. Yes or no?
Yes.
Because the whole protein synthesis machinery, which is ribosomes and other proteins and enzymes associated, all these are only evolved to recognize RNA and not DNA. So protein can only be formed from RNA, whether it is a prokaryotic cell or it is a eukaryotic cell. Proteins can never directly be formed from DNA. Okay, so write down. RNA can directly synthesize proteins. RNA can directly synthesize proteins in a cell and can express the characters whereas DNA depends on RNA for protein synthesis. Depends on RNA for protein synthesis. Second point, the protein synthesizing machinery of every cell has evolved around RNA. So even though DNA is better to store information, RNA is better for transmission of that information, transmission of the genetic information. Do you understand? Yes or no, people?
Yes, sir.
Yeah. Great. So write down, even though DNA is more stable, and a preferred and is preferred for storage of genetic information, even though DNA is more stable and is preferred for the storage of genetic information, RNA is better for the transmission of the genetic information. RNA is better for the transmission of the genetic information. Is it clear? Is this point clear? Everyone?
So clear.
Now, having said that and having discussed both DNA and RNA, you know that both are good at something. Now, if I ask you, which do you think, on the basis of everything that we have discussed, stability, reactivity, the ability to act as an enzyme, the ability to store information, the ability to transfer information better, structural and chemical stability, everything, keeping all that in mind, what do you think? Which was the first genetic material, DNA or RNA? RNA. Why do you think so, Atif? What is your rationale behind thinking that RNA was the first?
Because like RNA is way better for the transmission of information.
Way better for transmission of information. Okay.
Yeah.
So you, okay, so complete your sentence. Yes. So interesting. So what?
So for like earlier organisms, like primitive organisms, to transfer the genetic material, they should like started from RNA, something like that.
Okay, but you know, transfer can happen through DNA also. I just said that RNA is better because it can just directly code for proteins. If there is DNA also, DNA can make RNA and from DNA, RNA can be synthesized and then protein can be synthesized. It will just take one more step and it will be slightly slower. But I think early organisms had all the time in the world. They did not have to attend a 4:30 p.m. biology class, so they were never in hurry to do anything, right? So they had all the time in the world. They are early cells, no motives, no motivations, no ambitions, like as we have. So even if they had DNA, they can still transfer information. There is something that you are, again, think about it. There is something that only RNA can do and DNA cannot. And the answer lies there. Can you think? Aisha? Aisha, how are you?
Hello, sir. I'm fine. What about you?
I'm good. You were all silent and sneaky all this time in this batch.
Yeah.
Okay, cool.
I just came to India and
Yeah,
there's
I
no
know.
education.
So you'll be in this batch, right?
Yeah.
Very good. So you have to speak up. Why are you silent? Why everyone is silent? It's not like if I'm asking questions to Atif, only Atif is allowed to speak.
Sir,
Now the question here, yes,
there are
Aisha.
lots of people in my family and sometimes they will speak something random and that's why every time I have to be careful.
It's fine. At the end of the day, we all are studying here, right? It may happen, I always choose a background. I try to sit as close to the wall as I can so that no one moves from behind me. That's one strategy I do. You never know, in front of me all sorts of funny business is happening. Someone is walking around, someone is dancing around, so that we all are at home. And what we can expect, I can understand. That's completely fine. It should not inhibit you, or you should not hesitate because of that to discuss. You should discuss. It's fine. What will happen? Some person will say something randomly. It's okay. We all understand. Okay, so the question again is, Atif had some very good points on the table that we are discussing. He believes that RNA was the first genetic material, but he first gave me a reason which I kind of rebutted and discarded from my side, saying that even if DNA would be the first genetic material, what you are saying can still happen. The transfer of genetic information can happen. So I want something more concrete in defense of RNA, if Atif wants to stay there. And Asha, what do you think? Which was the first genetic material according to you? RNA or DNA? Aisha and Atif. Atif, you have to give me some better defense if you are defending RNA. Or you shift to DNA. If you defend RNA here, I am 200% sure whenever this question comes in entrance or boards, and it will come in some or the other form, you will not do it wrong because you actually defended it. You know what it means. Can you think of something, both of you, all of you, Fatima, Anifa, please come to the table for discussion. Can you think of some property of RNA which is only in RNA and must be very, very important for the early organisms that were evolving? The earliest cell or the earliest life form?
RNA can also be an enzyme.
Yes, very good, very good. So keep going. RNA can also be an enzyme. It can act as a catalyst, and we call those kind of RNAs ribozymes, right? Very good. So what? That is the question.
So maybe for early organisms to reproduce quickly, they needed the RNA as a catalyst.
Do earlier
I'm not sure,
organisms
sir.
always have to reproduce? Before reproduction, there are a lot of things. See, in the central dogma of biology, what comes first? DNA? And what comes last?
Yes.
Protein.
Protein.
But you are saying that there was a time when DNA was not present. Yes? And RNA was the first genetic material. I'm sure that you are not saying that DNA and RNA both were present and RNA was the first genetic material, right? If RNA was the genetic material, then DNA was not present at that time. Yes or no? That is the only possibility, because if DNA and RNA both will be there, the cell will always choose DNA because it's more stable. Make sense, everyone?
Yes.
So there was a time when RNA was the genetic material, the first genetic material, and what you are defending, Atif, is correct. So RNA has this ability to act. RNA also has catalytic properties, right? It can act as a catalyst. And when the first living systems were evolving, enzymes were not evolved, to do certain activities in the cell, certain very important and crucial activities or processes related to metabolism, splicing, translation, etc. You understand? All that thing was done by RNA itself. So RNA was a multi-specialty tool in the early cell. Now, if I tell you that I will only do one function and will do it the best, hire me. You are starting a new company, okay, Atif? And I came for a job, and that job is to manage something. The post of manager is vacant. And I came and I said that, I am a good manager. I'm the best manager actually. I have a lot of experience to manage. I'm very, very stable as a manager. Hire me. You are starting a new company, okay? Remember that fact. And then Aisha comes and says, okay, I do not have much experience in managing, but apart from managing, I can also handle accounts. I can also do some PR things. And I can also maybe, act as a HR for you. I'll hire also. Whom will you choose when you are beginning a company to work with you? You have limited resources, limited funds to expand. So whom will you choose? Atif, logically. You're there, Atif? Yes, you're not audible if you're there.
Sir, can you hear me?
Yes, who will be more beneficial for you? Could you hear me?
Yes, sir. I think it would be Aisha.
Yes, because she can do, like she may not be doing one thing with 100% of stability, but you know that she's multi special and she can do multifactorial things. And in the beginning, it is very important to get a company or get a cell up and running and become a stable life form, right? After that, you can then make different departments, assign one head to each department, an account department head and manager head and this and that, and then your company can shoot up from there. You understand, Atif? So that's the best part. So write down RNA. Okay, before you write down, so there is a concept in biology which now with enough proofs is validated and is called RNA world. At one point of time, it was a hypothesis. Now we accept it as a RNA world theory. So at one point of time, the world was governed by the RNA. The living world was governed by the RNA. So that's called RNA world theory. And this tells you that RNA was the first genetic material. Write down. RNA was the first genetic material. And why do we say that? Write down. It is because, or write down evidences. It is because essential life processes like metabolism, translation, etc. Essential life processes write down. Essential life processes like metabolism, translation, even protein synthesis, and biochemical catalysis. Evolved around RNA. Evolved around RNA. Okay. So RNA can act as a genetic material. RNA can act as a genetic material. And also as a biochemical catalyst. In the cell. In the cell. In the early cells or in the early living systems. Okay. And another evidence is that still, like in today also, in like in our cell, in animal cell, in plant cell, there are still certain very important reactions that are only catalyzed by RNA enzymes called ribozymes and not by protein enzymes. So now we have evolved all sorts of things. Now life form as of today have evolved a lot. It has a lot of tools, but still there are certain chemicals that can only be catalyzed by mRNA. Now, do you think that this is a fair enough evidence to suggest that RNA was the first molecule to gain importance, as it was, all rounder? You understand, everyone? Is it clear? Do I make sense? Yeah. Okay, so write down. Even today, there are some important biochemical reactions. Even today, there are some important biochemical reactions that are catalyzed by RNA only. By RNA only. And not by protein enzymes. Not by protein enzymes. So RNA as a genetic material was very good to begin with, right? Multifactorial, all-rounder. It helped the company, like which is life, startup. Okay, Atif. But as I said, after some point of time, you will need specialists, right? You cannot just keep running a company and
Yes.
keep. Yeah, the company can start, but it cannot grow without specialists and specific departments headed by specific people having specific roles in that company, right? You can no longer multitask when the things become more complex. Correct or not?
Yes.
So that's why DNA evolved from RNA, write down. But because RNA is more reactive and less stable, but because RNA is more reactive and less stable, DNA has to evolve. DNA has to evolve to become the genetic material. Okay. And DNA has one more special property. There is something called repair. DNA is not only more stable, but it also has one mechanism of proofreading. So during a process of replication, if a DNA, or in general, if a DNA gets any damage, a wrong base pair added, a cut in the DNA, DNA can repair itself. It tries to repair itself in the cell. It has mechanisms evolved that repair the DNA. That's called DNA repair mechanism. Okay, write down. In addition, DNA has evolved. In addition, DNA has also evolved a DNA repair mechanism. A DNA repair mechanism. Okay. So from RNA world, we now come to DNA. And the first thing we will talk about of the DNA is replication. Do you understand, everyone? RNA is clear?
Yes.
Still, like today also, there are many viruses that have RNA as their genetic material and not DNA. Do you understand?
Yes, sir.
But because DNA evolved for some special reasons, so let's talk about it now. So from RNA world, we now come to DNA. So write down replication. So what do we mean by replication? Once tell me. Quick. Simple. What does the word replication means?
The ability of DNA to create copies of itself.
The ability to DNA to create copies of itself. Very good. Now, how do you think DNA creates copies of itself? So DNA has a double helix structure, right?
Yes.
So this replication mechanism was also proposed by Watson and Crick, because when they realized the structure of DNA, they immediately also realized and figured out how will DNA replicate, how will DNA be formed from pre-existing DNA. Okay, so since DNA is a double helix structure, so let me make a double helix structure. So let's say this is one strand and this is another strand. Okay, this is the double helix structure. Now this strand opens. Okay, first has to open from each other, and they proposed that a new strand is formed complementary to each DNA strand. Do you understand, everyone?
Yes, sir.
This way, so this is known as the semi-conservative DNA replication. Why it is called semi-conservative? Because you see these black ones. If I let me make it a little more clearer so that you understand. Give me some time, people. So do you understand that this black is the strand, right?
Yes, sir.
These are the bases. Then after one point of time, it opens. And now there are two strands. You understand, all of you? So they are
Yes, sir.
basically the strands Yes. So these two are the original strands, what is shown in black. And when a DNA replicates, what happens is a new strand is synthesized. Do you understand? Which I have shown
Yes,
in
sir.
blue. But when this is happening, so now there are two molecules, right, of DNA.
Yes, sir.
Yeah. Let's see like this. Okay. Now, can you see that in these two strands, which are formed from this one strand, basically, it first opens, but in both these two strands, half of it is new, but half of it is older only. It's the older one itself, right?
Yes, sir.
Now, so this is called DNA replication fork. What it is called? DNA has to open up before replicating and act like a fork. Can you see? One thing becomes two. And because 50% of the new strand is actually the old strand only, this type of replication is called semi-conservative. What do you mean by conservative? Conserve. What do you mean
Conserve
by
the
conserve?
old.
Yeah, conserve means to keep what you got from like behind or before or from your old people, right? So semi-conservative scheme or semi-conservative method of DNA replication, it's known as. Do you understand, everyone?
Yes, sir.
So write down. The semi-conservative method of DNA replication states the semi-conservative method or scheme of DNA replication states that the two strands would separate, that the two strands would separate and act as a template for the synthesis of new complementary strands. Do you understand this?
Yes.
So now technically, if I ask you, when these two strands separated, just follow the black one, the original. So let's say this is the original 1 and this is the original 2. So this complementary, I'm marking it as A. The complementary formed by from 2 is, no, let's say this is A and this is B. Okay. Now A will exactly be like 2, right? Because before opening, 1 and 2 were complementary to each other. Do you understand? 1 and 2
Yes.
were paired with each other. When 1 and 2 opened, the black ones, and new blue ones, I'm just coloring it different to explain you, and the new blue ones are synthesized, then the strand A which is marked, can you see A? Complementary strand of 1 is A and complementary strand of 2 is B. Everyone on the same page? Fatima, Aisha, Hiba?
Yes, sir.
Yes. So the complementary strand to 1, which is A is same as 2, right? Yes or no? And B is same
Yes,
as
sir.
1, correct? They are exactly same.
Yes.
The sequences are also same. Now, this is called semi-conservative method. Now, how to prove it that what they said was true? Because they just thought this is the most logical explanation to explain how DNA must be replicating. But how to prove? So where did the experimental proof came from? Now, this is a very, very important part of this chapter. The experiment that we are going to discuss now is regarded in biology as the most beautiful experiment ever conducted in biology, okay, and which is known as Meselson and Stahls experiment. So the experimental proof came from Meselson and Stahl. There were two scientists. Stahl's experiment. And this experiment is known as the most beautiful experiment in biology. It's regarded like that. Now, this semi-conservative was proposed by, write down, it was proposed by Watson and Crick. This part, okay. But was proved by Meselson and Stahl. You understand? Please draw this and write, then I'll move forward with the experiment. This experiment was done in 1958. Done, people. Shall I move forward? Shall we start with the experimental proof? You understood the semi-conservative method of DNA replication. You understood the scheme. Now we have to do the experiment. Rifat is done. Others? Yes, Atif? Okay, very good. So now let's begin. So what they did was, they had an idea. Now to make that idea work, all they had to do, so they took bacteria, E. coli, because in E. coli, you know that there is only one DNA, right? One chromosome, which is like one DNA. It's a bacteria, so it's prokaryote, it has just one DNA thread, okay? And when bacteria divides, it replicates the DNA so that it goes into the next generation? Yes or no? So from one thread,
Yes, sir.
there must be, so from one polymer of DNA, now there will be two polymers made. Yes. So first what they did, they took E. coli bacteria with normal DNA. Now you know that in normal DNA we have nitrogen also. Okay. So this nitrogenous base, remember I told you there are 4 nitrogenous base. So nitrogen is an important component of those bases. Yes. Now this time what they did, if this semi-conservative scheme is correct, then they took E. coli, then they grow the E. coli, they cultured the E. coli in a heavy isotope of nitrogen. Now remember, they did not use radioactivity here. They are just using a heavy isotope. So isotope means same atomic number, different atomic mass, right? What is the normal atomic mass of nitrogen? 14, right? N14. Is it clear?
Yes.
Atomic number of nitrogen is 7 and atomic mass of nitrogen is 14. So they took a heavier isotope which is called N15. In N15, there are 7 protons in the nucleus but 8 neutrons. So the mass becomes 15 instead of 14. So it is not radioactive, it is just a heavier isotope. So it is not going to affect the cell or the DNA in any way through radioactivity. It will be normal. But now what happens is, imagine. So I will use black, let's say for there is this E. coli bacteria in which there is a normal DNA. Okay. And I'm representing that DNA here. Okay, everyone.
Yes, sir.
So their idea was, this is the normal DNA containing all the nitrogenous bases will be containing nitrogen 14, right? N14. Yes.
Yes, sir.
So if you take normal bacteria, E. coli, and if you start culturing it for like generations in no other media, but now the culture media contains N15. There is no N14 available now. Okay. So what will happen? This strand will open for replication, right? So
Yes, sir.
this was normally like this, but to replicate, it will open, right?
Yes, sir.
So this part still contained normal nitrogen. But now let's say when the strand is forming, the next strand that will be formed, let me make it a little better. In this case as well as in this case. So when this new strand will be formed, it will be containing all the bases will have nitrogen 15, right? Not nitrogen 14, because there is no nitrogen 14 to make new bases. So when we replicate DNA, we make new things, raw materials, right in the cell. And where does those raw materials come from? From our nutrition, from the food, from the environment sources, right? Understand, everyone? Say yes or no.
Yes, sir.
So in the next generation, after one division cycle, if you only have allowed the DNA, then you will see that the two strands of newly formed DNA after 20 minutes. So this is at 0 minute, like they are culturing, and then after 20 minutes, which means why 20 minutes? Because after every 20 minutes, E. coli divides. You know that? So one strand will contain nitrogen 14, the normal one, and the other strand will contain nitrogen 15, right? The newly formed one. Because the only nitrogen present now is, so you will see a hybrid strand, right, in the next generation, if this is true.
Yes, sir.
Now tell me, so this is a normal strand, original strand. This is a hybrid strand. Both these two strands are hybrid. Now, hybrid will be denser or heavier than the normal or lighter than the normal?
Heavier than the normal.
Heavier, right? Because half of the strand has a heavy nitrogen isotope everywhere. Is it making sense?
Yes, sir.
And what will happen after 40 minutes? After 40 minutes, what will happen? This strand, which is already there, it will open. Oh, sorry. This strand, which was already there, will open, right? The hybrid strand was there, and that will open. Because after 40 minutes, again the bacteria will replicate, yes or no? And this time, what is present in the system, like in the environment? Again, nitrogen 15. You are continuing to grow it in nitrogen 15 only, right? So again, one of the strand will be hybrid, but the other strand will be purely nitrogen, right? Based. Does it make sense to you? People, does it make sense, yes or no?
Yes, sir.
So here you will see two strands, both of them. Let me just complete it first. It's difficult to dictate while drawing. Yes, please also, you also draw it with me. Okay. Now we started with a hybrid. All were hybrid, right? Remember? And now this strand is hybrid, you know, containing nitrogen 14 and also nitrogen 15. But this strand is having nitrogen 15 in both the strands, right? This DNA. So this is a heavy DNA, a normal DNA, hybrid DNA, and this is a heavy DNA. You understand?
Yes, sir.
Now, after 40 minutes, there will be heavy DNAs that will start coming, right? After 80 minutes, there will be more heavier DNAs, correct? So what will happen if, and this experiment was very, very beautifully conducted. So let me just give me 1 minute. I'll explain. Please draw this thing first. You understood what is he doing? He is culturing E. coli in heavier nitrogen isotope medium. both of them. You understand? You can still see the screen,
sir.
everyone? Yes or no? Yes, just give me 1 second. Let me pull this figure for you. You all have NCERT with you? Please
No, sir.
open the NCERT. No? Okay. So let me pull. Those who have NCERT, please open your NCERT. Pulling the exact figure. Yes. Yes. Can you all see this figure? This is from your NCERT.
Yes.
Page number 05. Let me make it a little bit. Is it clear now? Clearly? It's visible clearly?
Yes, sir.
Now let's see what's happening here. This is Meselson and Stahls experiment. So as I told you, they started with nitrogen. So what they did here is they cultured the E. coli. So we are going. So you understood when we will start culturing the E. coli in a N15 media. After some time, we will get like we will get all the bacteria which will have heavy like strands, which will have all the nitrogen 14 replaced by nitrogen 15. Yes. So first you have to culture the E. coli for so many generations that all the nitrogen 14 gets replaced by nitrogen 15. Okay, clear? So this is what they did. They reached till here where we also reached. So we started with the normal N14 E. coli. We cultured them in a media containing N15 for many generations. And after many generations, all of them, after 40 minutes, at least like one bacteria will have the DNA, one DNA completely made up of nitrogen 15. And if we keep doing it, so all the hybrids and the normals will die. Okay. And we will be left with heavy isotopes. Is this clear, everyone? And this is where we will start the experiment. So if all the E. coli have N15, so the red mark is for nitrogen 15 DNA. Do you see it here? If you keep these
Yes,
E.
sir.
coli in a test tube and centrifuge the test tube, like you took all the DNA, you removed, you took all the DNA out, so that's called DNA extraction. And if you centrifuge the tube, there will be a band, right? N15, N15, where both the strands have nitrogen 15, there will be a band which will show up, right? Then you allow the bacteria to grow in normal environment. And in normal environment, what is the normal isotope of, like what is the normal nitrogen present? Nitrogen 14, right? In the atmosphere or everywhere around us, nitrogen 14 is present. Is it clear?
Yes, sir.
So the first generation again will have one strand made of nitrogen 15 and the other strand made up of nitrogen 14, because now you are reversing back. So whatever you did till here to reach N15 heavy DNA, it will automatically get reversed now. So in the first generation, which is after 20 minutes, you take the DNA again. For all the bacteria, you will see that the band is coming a little, like the band has shifted a little bit towards the tip. So it was here at position A. So this was position A. Now there is no band at position A, there is a band in position B. You understand why it happened? Because the
So-
DNA now is getting lighter. Just in the manner it started getting heavier. You understand? So it got heavier when we cultured it in nitrogen 15. When all of them are heavy, then we did the opposite. We allowed them to grow in normal environment. So in the first generation, all will be hybrid, which will be lighter than the heavy. So hybrid is always lighter than the heavier DNA. And after 40 minutes, which is like generation 2, you will get, so they will also open, and now there will be 4 strands, 1, 2, 3, 4, and 2 of which will be totally light, and 2 will be hybrid. You understand?
Yes, sir.
So again, you will see that the band which you got heavy band at position A, again there is no band at position A because it's semi-conservative. You will still see some band, a light band at position B, and another band which come up at position C now, which will be even lighter than the hybrid one. You understand? So what does this proves? This proves that with every replication, the DNA is opening, the double strand is opening, and a new strand is forming. This is why, this is the only way through which you can explain why the heavier DNA with which we started the experiment, or the lighter DNA with which we started the culturing, it disappeared right in the next generation. Where did it go? Where did this band go? Why is it not there? If DNA would have replicated in any other manner apart from semi-conservative manner, then this band would have been there, right? Because the original DNA would have been conserved, fully conserved. So it's not fully conserved, it's semi-conserved. One strand goes to one DNA molecule. That's why the band is gone. There is no heavy DNA after the first replication because both the heavy strands open and then the light strand is formed around them. So both becomes hybrid strands. Making sense, everyone? Is it clear?
Yes, sir.
Yes. Please draw and mark this. Let me know when you're done. So this is the experiment. Now let's write something about it. Are you done? I'll dictate the experiment, what you have to write in your exams. Let me know when you people are ready. So when we apply gravitational force, this is the point. The substance which is heavier will go towards the extreme end, right? So at
Sorry,
this place, when we apply gravitational force and rotate something, heavier substances goes towards the extremity, right? They shift. Heavier the substance, farther it moves away from the center. Yes or no?
yes, sir.
This is the center and if I'm applying a gravitational force and rotating it, then if something is 10 grams, it will move farther in that medium. If something is 5 gram, then the lighter the substance, the closer it is towards the center. You understand? So the lightest chain N14 will be closest towards the center. The hybrid chain N14 will be at an intermediate distance from the center, and the heaviest chain N15 will be the farthest from the center. Clear?
Yes.
Yep. Let me know when you're done. So you can actually see beautiful bands in the test tube where you are doing this experiment after extracting DNA from this E. coli that you have cultured. So you just culture E. coli in a nitrogen 15 medium for many generations, allow all their nitrogen 14 to get replaced by nitrogen 15. Then you start the experiment, you take out that nitrogen 15 E. coli, allow it to grow on a normal medium, and after every generation, extract their DNA and see where they lie, where the band comes up in the centrifugation experiment. And if you see a heavier band at a different position and a medium band at a different position and a lighter band at a different position, you will come to know that there is a semi-conservative method of replication. Yes?
Yes.
Okay. Let's start. Write down. First point, Meselson and Stahl. Meselson and Stahl cultured E. coli. Cultured E. coli bacteria in a medium containing NH4Cl. Where did I keep it? Medium containing NH4Cl. Anyone can tell me what is NH4Cl? What is NH4?
Ammonium chloride.
Perfect. It's ammonium chloride, but this N was all nitrogen 15. It was a specially prepared NH4Cl. So it was a heavy ammonium chloride. So they cultured E. coli because something has to give nitrogen to E. coli, right? To use it. So they cannot just give gaseous nitrogen, no? So they give ammonium chloride as the only nitrogen source. As the only nitrogen source. So they made sure there is no other nitrogen source. They are culturing it as in closed chamber so that atmospheric nitrates and other things cannot go there. So the only nitrogen source that the bacteria can use is ammonium chloride with nitrogen 15 for many generations, as the only nitrogen source for many generations. This finally led to next point. This finally led to the replacement of nitrogen 14 with nitrogen 15 in the DNA of newly synthesized bacteria. In the DNA of newly synthesized bacteria. Now this heavy DNA N15, this heavy DNA N15 could easily be distinguished, distinguished from the light DNA N14 N14 from the light DNA by centrifugation. By centrifugation. And you know, this kind of centrifugation has a name. It is called cesium chloride density gradient based centrifugation. So this centrifugation that was done, it is a special centrifugation. It's called cesium chloride CSCl. Okay, this is the short, this is the formula, CSCl. Cesium chloride density gradient based centrifugation. This was done. Okay. Write down. Next point. And also remember, it is not a radioisotope. Like, you know, we used in Griffith's experiment. No, not in Griffith. Which one? Martha Chase. In Martha Chase experiment, Hershey and Chase's experiment, we used a radioactive isotope of sulfur and phosphorus, correct? To distinguish between protein and DNA and to give a proof that DNA is the genetic material. Here, we are not using radioactive nitrogen. We are using a heavy isotope of nitrogen. Simply, it is just heavy so that it can be separately distinguished through centrifugation. So write down. Next point. After many generations, the E. coli cells were transferred after many generations. You know, basically when all the DNA was replaced by heavy nitrogen. So after many generations, the E. coli cells were transferred to a medium containing normal NH4Cl, where the N is 14. So just write this. So this was the heavier NH4Cl. And from here onwards, they started giving the normal NH4Cl. So NH4Cl, ammonium chloride. Now it is 14. Okay. Clear, everyone? Okay, write down. They transfer the cells into the normal ammonium chloride, NH4Cl, and 14, and took the DNA samples after every cell division. After every cell division. The DNA that was extracted, next point, the DNA that was extracted. I hope I'm audible, right? Everyone is writing?
Yes, sir.
The DNA that was extracted from the culture 1 generation after the transfer. The DNA that was extracted from the culture 1 generation after the transfer. Basically this one. You can also write in brackets 20 minutes. Had all the DNA strands of a intermediate density. So to begin with, it was a heavy density. Then after 20 minutes, all DNA strands were intermediate density. And after 2 generations, there was a equal proportion of intermediate density DNA and light density DNA or normal density DNA. Now, can anyone, now this question is for, let's say, who's there? I have a question for Fatima. Fatima, are there? Can you speak? Fatima? No? Hanifa, are you there? If you understood this experiment, you have to tell me. Are you there?
Yes, sir.
Yes, Hanifa. You understood the experiment? No problem?
Yes, sir.
Yeah. So tell me, what do you expect after 80 minutes? After 80 minutes. So write down this question. This is, I think, also a question in your NCRT. What happens after 80 minutes? In this experiment, the Meselson and Stahls experiment. So if I take the DNA sample after 80 minutes, what will be the expected, like what will you expect? The proportion of DNA, or you have to answer in that. Hanifa. Tell me. What will be the proportion of light and hybrid density DNA molecules? That is the question. Hanifa? Hiba? You're there? Hiba and Fatima both are not speaking. Hanifa is also not speaking. Rifat, are you there? Can you tell me what happens after 80 minutes? Yes, please. Rifat, if you're speaking, you're not audible to me. I can hear something, but not your voice. Are you all having some network issues? I was expecting. Nifa is
No,
having
sir.
issues. Okay, Atif, then you answer. What do you expect after 80 minutes? what will be the proportion of light, intermediate, and heavy DNA? Will there be any heavy DNA?
No.
No. So there will be no heavy DNA. Remember, the DNA, if you start with a heavy DNA and start culturing in normal nitrogen, there will be no heavy DNA strand right away from the first generation. That is the rule of semi-conservation method. So what will be the proportion of light and intermediate DNA? Tell me how many, if I start from one, if this experiment continues. So we started with 1, it became 2, then it became 4. At 80 minutes, how many DNA strands will be there? How many of them will be light and how many of them will be intermediate?
So there will be 16 DNA, 8 of them will be light, 8 will be intermediate.
8 will be light and 8 will be intermediate, you're saying. So let's go to the answer. So if I started with 1 strand, then after 20 minutes, there was 2 strands. And after 40 minutes, there were 4 strands. So after 60 minutes, there will be 8 strands. And after 80 minutes, there will be 16 strands, right? 16 DNA molecules. So this is correct, right? Very good. Now you said, what was your answer? 8 will be?
Sir, I'd like to change my answer.
Okay, allowed. Change your answer. And now you have to tell us what mistake did you make if you are changing your answer. Yes, change your answer quickly.
Yes, sir.
What is
I
your-
always thought it would be in equal proportion, but that does not seem to be the case.
Yes, so what will be the case? How many, tell me how many light strands and intermediate strands will be there, like molecules? How many light DNA molecules will be there?
Sir, I think 14 will be light and 2 will be intermediate.
2 will be intermediate.
Yes.
So there'll be no more intermediates. 2 intermediates were there right from the first generation. Then in second also there are 2. You are saying in third also there will be 2. In fourth also there will be 2.
Yes, sir. I think so.
Yes, you think correct, because see, to make an intermediate DNA, you need to have one strand which is nitrogen 15, right? There were only 2 strands with nitrogen 15 because from there onwards you started giving nitrogen 14. So these strands will remain as it is, and when they will open, they can maximum form 2 new molecules, right?
Yes,
And both
sir.
of them will be intermediate. Then again they will open, but because heavy chain is not being formed anymore, so you are true that there will be 2 intermediate molecules and 14 light molecules. If I ask you after 100 minutes, what will happen? Again, there will be 2 intermediate molecules and 30 light molecules. You understand?
Yes.
After 120 minutes, the total is 64. There will be 2 intermediates and 62 light. Everyone understands this? This is why they asked you this question. Everyone understands the answer? Please say yes or no. At least write yes or no. You understood, Atif?
Yes, sir. I understood.
Very good. So you'll not forget it now. Write down after 80 minutes, total DNA molecules will be and you don't even have to answer in terms of total DNA molecules. They ask you the proportion. So what is the proportion, Atif? What is the proportion after 80 minutes? 2 intermediate and 14 light. So what is the proportion?
The proportion is like the ratio.
Yeah, you can tell the ratio. Tell me the ratio. 14:2, right?
Yes, sir. So it'll be 7:1.
Or I
1
can
is to
say
7.
yes, 7:1. Or I can say the light DNA molecules will be 7 times more, right?
Yes, sir.
Yeah. So heavy. It's not heavy. There is no heavy. Intermediate to light DNA. 7:1. Understood? Why are we
Yes,
not
sir.
giving the exact number? Because the question has not asked us how many bacteria did we begin from. Okay, please fix your mic, because see, the point is, if you people are not discussing, then I cannot help you beyond what I'm teaching you. So if I ask you multiple times, do you understand? Do you understand? Because you need to understand. I'm not teaching for myself. I know these things. If you do not know anything, then we should not move forward. Is the motive here? Okay. Please try to fix it so that all of you can participate. And it's the more people participate in discussion, the better it becomes. And also I get motivation to teach. Okay, very well. So all of you understood all these experiments? Now, next thing that
Yes,
we
sir.
have to do in this chapter, we realized that DNA is the genetic material. We also realized the structure, and we also realized how DNA replicates. So we are started with replication, and we realized that it is semi-conservative method of replication. Now, the next point, the next thing that we have to understand is the machinery and the enzymes. So it is very, now we are going into hardcore biochemistry, like molecular biology and biochemistry. So machinery and enzymes involved in replication. Okay, so we'll do the same for transcription and translation as well. Okay, now talking about machinery and enzymes, I'm giving you how much time do we have, or we do not have much time. So let's do one thing. You understood the replication fork, everyone?
Yes.
So let me give you some homework. So your homework is, this is a DNA. Now I will not make from here onwards, every time I will represent DNA, I will not make that helical structure. You know that it is helical. For the sake of convenience, I'm making it like this. And we know that it opens, right?
Yes, sir.
When it opens, the new strand which is formed, so in one direction, it is continuous. Okay. So it forms continuous in one direction, the new strand, but it forms discontinuously in the other direction. You understand? So this is the DNA.
Yes.
This is the replication fork. At one side, it is continuous, and on other side, it is discontinuous. So what you have to go back and read about is first the replication fork. Replication fork. Try it from NCERT and try to understand why one is continuous, one is discontinuous, along with one more concept, which is origin of replication. Origin. And when you are doing this origin of replication, you will come across a word, whether you are reading NCERT or any other book, you will come across a word called DNA polymerase. This is the hero. Okay. This is the enzyme. Remember ASE. Whenever there is ASE, peptidase, lipase, amylase, anything. Okay. Hydroxylase, polymerase. We know that it is an enzyme, right?
Yes,
This is the
sir.
replication enzyme. So try to figure out something about this enzyme as well. So this is your homework for today, because I'll not start this. It's a long topic and we'll try to finish replication and begin with transcription. Transcription is shorter as compared to it. And I think we have things in transcription. So in the next class, I'll try to finish replication, transcription, and translation. So this is your homework with respect to replication. Okay, noted. Everyone has noted down what you have to do. If you have any doubts, anyone has any doubt or any question, please ask me. I'm not starting the new topic. I'm not starting the machinery and enzyme today. We'll do it tomorrow, like in the next class. Any doubts, people?
No, sir.
No doubts?
No, sir. No doubt.
Okay, so let's declare it a little early today, few minutes early today. Please read. I'll advise you first read from NCERT, and if you do not understand the language, because sometimes NCERT writes very concise, you need some elaborative. So you can refer to other books as well to understand the concept. Okay, if you cannot understand it in a very concise manner, which is present in NCERT, but do read this thing. Okay, and then come, please. Okay, Atif. I
Yes,
hope
sir.
you will do this. No traveling this time.
No, sir. Problem.
Cool. Okay, then see you in the next class, people. Take care. Bye bye.
Thank you, sir.
Okay. Bye bye, Refat. Take care. Please fix your mic.